All right, cool. So yeah, thanks everyone for coming. I'm really excited for today's presentation. Dr. Dwyer was not only one of my professors here at UB, but also is doing really cool neuroscience research that I'm very interested in. So let me give you a little bit of background about Dr. Dwyer. So he's an associate professor of neurology and biomedical informatics here at University at Buffalo. Received his undergraduate education in information technology at Excelsior College before doing a master's in computer science at Nova Southeastern University. I never actually knew that, Dr. Dwyer, that you had the computer science background. Then he took a trip across the pond for his PhD at the University of Bradford in England.
Yep.
And as of today, he has published exactly 167 research articles with over 7,000 total citations. He's an expert at developing neuroimaging techniques, and we are extremely grateful to have him here to present for Ask. There will be a short Q&A session at the end of the presentation, so if you have questions, please save them. And Dr. Dwyer, go ahead and take it away.
Thanks, Ben, and thanks, Stephen, for making this possible. It's really nice to be able to talk to you all. So as Ben said, my name is Mike. I'm a researcher with a kind of interesting background. I really got started in computer science and computational stuff, and then got really interested in the medical field and moved into looking especially at neurological disease. A lot of focus on multiple sclerosis, but other diseases as well. I think it's pretty impressive what we can do these days, combining computational tools with really neat methods like MRI. And it's, Ben and Stephen said there's a lot of different people from different backgrounds here, so I'll try to keep the talk at kind of a medium level and hope it's a little bit interesting, or there's something for everybody. And definitely happy to answer questions at the end. I think it's a really cool time for you guys for everybody who's involved in science or getting involved in science to be doing these things. I mean, the pace of discovery is incredible. There's so many new things created every day that it's just wonderful to have this kind of interest in science. So I'll try to tell you about a couple of the areas that I research in a kind of broad way and just kind of give you a survey. One of the things I'll give you a little bit of background in is brain atrophy and some of the tools we've been developing to look at that in diseases. Some of the things we've been doing to do tissue measures of disease, so we can use MRI imaging to look at how diseases are progressing in the brain. And then some really interesting stuff with structural and functional connectivity that we've been doing with some more modern MRI techniques. I'm not sure why there's the figure. But before we talk about that, I know we have a pretty broad spread of people. So, I just want to touch on what is MRI, magnetic resonance imaging, because it's a really, really, really cool technology. That's the stuff that was kind of, it was a Star Trek idea decades ago, and now it's a kind of commonplace reality in medicine. But essentially, we're using superconducting magnets to build tools that you can put someone in and image in vivo. And so that on the left is what a conventional MRI scanner looks like. You get on this bed, and I'm sure some of you have probably had one at some point in the past, and you kind of go in this little claustrophobic tube, and you hear a bunch of sounds that are just gradient coils going, and 10 minutes, 20 minutes, an hour later, you have beautiful images. And what's really neat about MRI are a couple things. First of all, it's what's called a tomographic imaging method. So that means it's not like X-ray where we just get one picture. We get that's kind of like a silhouette. With MRI, we can really get individual slices of anatomy, almost like you would get if you were to slice into a brain in a pathology lab. And so you can really see this kind of beautiful detail in all kinds of different angles. It also is really nice because you can build that up to get true volumetric imaging. So again, unlike something like an X-ray, it's not a shadow on the wall. We really can see the entire image, and you can build up the full three-dimensional representation. And I think a lot of people know that, kind of thing about MRI nowadays, but one of the best things for me as a researcher, one thing I really love about MRI as a modality is that it's actually not just one modality. It's really a multimodal thing. And what I mean by that is when I think the best analogy is kind of the different kinds of light that we have. We have ultraviolet light, infrared light, and there's some really interesting stuff. If you look at flowers under different lights, they look totally different. Insects see flowers very differently than we do because they see those wavelengths. And MRI essentially can also look at different aspects of the same anatomy. So we don't just get one picture when we do an MRI. It's not like when we go in a CT, for example, where you get one picture about how much does X-ray pass through that tissue. Instead, we can get 5, 10 different types of images that are maps of the tissue we're looking at, and they all tell us different things, and they tell us very interesting things that can be used in lots of different ways to really understand what's going on in the anatomy. Of course, it's not as perfect as having a histology workbench and looking with a microscope, but for something that is totally non-invasive, that we can do in vivo repeatedly and is totally safe, it's pretty impressive that we can look at and see all these different kinds of details. So very, very neat technology. All the magic happens from the protons in your body when you go in a scanner. And we don't have time now to go into all the details about how it works, but it's interesting that essentially when you go in a scanner, the scanner just kind of talks to the protons in your body and asks questions. And so that's how those different modalities can come about, because you can ask different questions. And just so you understand what MRI means, it stands for magnetic resonance imaging. And again, this is kind of what it looks like in a schematic view. You guys can see my cursor moving, right?
Yep.
Okay. So the magnet is a superconducting magnet. Again, it's 3 Tesla. And just to put that in a kind of scale for you, a single Tesla is 10,000 Gauss, and a refrigerator magnet is about 100 Gauss. The Earth's magnetic field is about 1. So this is, a magnet that's about 30,000 Gauss. They're incredibly powerful magnetic fields. If you walk in and hold a belt out, it'll stand sideways and get pulled to the magnet. And the real core of it is that magnet, these things called gradient coils, and an RF coil that's basically just radio antennas. And what we do is we put you in a strong magnetic field that lets us kind of isolate different pieces of your body and talk to different protons. And we energize the proton using what's called a resonant frequency, so that we can kind of communicate on a special channel with that with a specific set of protons in a specific little area of the body. And we ask it a question like, how much water is there in your local area? How free are you protons to move around? How often do you bump into other ones? And we can ask those kind of questions in using some sophisticated tools that people have worked out over a long period of time. And then we can do that in all kinds of different places in the body, one at a time, and kind of we can build up an image. I know this is a little grotesque to imagine, but it's imagine that the body's built of tiny cubes, and we can ask that question at each cube, and that's kind of the imaging component. And so we have this MRI that lets us build up these images. So, what are some of the basic applications of MRI? These are what kind of conventional images look like. And some of the ones that are most common are these T2 and flare weighted images. And one of the things that's worth noting about MRI is that a lot of times the kind of contrasts we get are not that not as obviously macroscopically meaningful. So when we measure T2, we measure something that's called a spin-spin interaction, which has to do with how quickly protons can give energy back to each other or interfere with each other. Or we measure T1, which measures how quickly they can relax back to a field. And I know that means nothing to most people, but we learn over time that those parameters are associated with certain things. in the same way that red wavelengths of light, we learn are associated with red things, and we can interpret that. So T2 weighted imaging tends to be very sensitive to changes in water content. And so we can use that to look at pathology, for example, in multiple sclerosis, as in these images here. And FLAIR is also a T2 weighted type of image that we use a special trick to null out the CSF, the cerebrospinal fluid in the brain. And we can start to really see lesions and pathology. And so you can use this kind of technique to find all kinds of problems in the brain and in the body. And it's been very powerful over the last few decades. It lets you find stuff in a much more nuanced way than you can find with something like CT or more classically X-ray. We do have injection methods in MRI still. You can inject things like gadolinium contrast that let us image, for example, the breakdown of the blood-brain barrier. So these are contrast agents that have a magnetic component that can affect the magnetic field. And when we inject that and it leaks into a certain area, we can see that the blood-brain barrier is less intact in that area. And we can see, for example, gadolinium lesions, or we could also use it to look at blood flow and perfusion in the brain. And those are contrast enhancing lesions. And we see this in a number of neurological diseases. They're very common in multiple sclerosis, for example, where we see that breakdown of the blood-brain barrier. Another type of imaging is that T1 type we talked about briefly, that can let us look kind of at macromolecular content a little bit, not as good as some like MTR, but it's a kind of classical method that's used and we can see kind of neurodegeneration a little bit. And we can also see, we can also use it for very high resolution imaging techniques that let us look at anatomy. So if you suspect, from a clinical perspective that there's some misfolding of the cortex somewhere or some dysplasia, you can see that using these techniques. The last conventional one I want to mention is diffusion MRI. So diffusion MRI, and we'll touch on this a little bit later, is a technique that's sensitive to how much water can move around. And not move around, I need to be careful, to diffuse. It's a microscopic phenomenon. So it's not like there's water sloshing around there. It's more about how free it is to diffuse. But this is a classical CT image. And these are images taken around exactly the same time that are looking at stroke. And you can see how much more sensitive MRI can be to see these kind of areas of lack of blood flow or infarction, ischemia of the tissue. And I think, that image speaks for itself, and it's really become a major thing in stroke. So those are some of the conventional things. MRI overall can be used to do lots and lots of things, and there's lots of different areas of research. Something we'll touch on is you can use it to track white matter fibers. You can use it to quantify concentrations of metabolites. So there's tools called MR spectroscopy. If any of you have done or do spectroscopy, in a lab, we can do the same kind of thing using MRI in different areas of the brain or other tissue. We can quantify iron content. We can look at vascular flow. You can look at regions that are activating and how they coordinate. So, bottom line, MRI is it's a really versatile technology, and I think a really cool accomplishment for science in general, that we've been able to build these kind of tools over time. So from my perspective, what's been very interesting is how we can marry, computer science approaches and digital image processing tools with MRI, to move from a kind of purely clinical zone where people are just looking at an image and saying, oh, it looks like there's some pathology there, to instead turn those images into quantifiable scientific data, and to turn that data into information that we can really use. And there's a lot of different tools there too. I imagine some of you are probably interested in these kind of things too. Digital signal processing, there's things like pre-processing and filtering, feature extraction, tools to co-register images, things like pattern recognition. The newer stuff, computer vision is a bit of a higher level about trying to understand images in a kind of deeper sense. So especially AI and deep learning. I'll try to show you a little bit of that. object identification, looking for things like pathology, tumors, lesions, or even quantifying normal objects that are in the brain to identify whether they're changing shape or size, and of course making predictions from images. So all these things are things you can do when you start to put those together. So one of the main areas I've done a lot of work personally is in atrophy, and we've understood now in a lot of diseases that we often look for these kind of obvious changes, bright areas, lesions, things like that, but we often miss the forest for the trees. And we've learned in a lot of these diseases that atrophy, loss of brain tissue over time, can be a really important marker of what's going on and a predictor, and it can help us understand some aspects of the disease too from a scientific perspective. So we see that in Alzheimer's, in epilepsy, in Parkinson's, we see it in ALS. We've especially seen it a lot in multiple sclerosis, and it's becoming an endpoint in a lot of the clinical trials that are done in this field. And so, to do that, what we do is we take these three-dimensional images and we use computational tools to identify and classify each pixel in the image as belonging to one of the major categories of brain tissue. We have cerebrospinal fluid here, you can see in blue. We have gray matter in green, and we have white matter in red. Sorry for saying that gray is green and white is red and all the different color things, but essentially we can do that and then actually quantify. And instead of saying it looks like there's a little bit of atrophy from a radiologist, we can say this person's brain is 1.536 liters, and then we can follow it up over time and do that again. And so putting these tools together lets us quantify things. And I think that in science, quantifying is a very powerful tool. It lets you do a lot more sophisticated measures. It lets you move to statistical comparisons, and it lets you really put things in context. And we have other techniques that we work on to, for example, parcellate brains into different structures. So we use computer vision and image processing techniques to not just classify and quantify the whole brain, but to identify known regions of the brain. So I don't know how many people here have a neuro background, but even though the brain looks like a big unified mass, it has a lot of different eloquent areas that do very different things. And those different regions can respond differently in different diseases. So we have computational tools that let us look at those areas. So we have precentral and postcentral. These are your motor and sensory areas here, and we have things like auditory cortex and visual cortex. And if we can do these kind of quantifications, we can understand better how they are changing in disease. We can do that in groups. I'll skip that slide for now. One of the things that we've worked a lot on, and I guess I would ask from aspiring scientists too, especially, and one of the things that has been very nice to see over the last decade or so is a huge push toward what's called translational research. Basic science is incredibly important, and I would never say that we shouldn't be doing it. But I think it's important that we also try to make sure that the findings that we discover are translatable to real world clinical situations. And so we've worked on a lot of techniques that don't just work on kind of ivory tower academic images, but that you can use more robustly in a real clinical setting. And so we developed, for example, a tool called Neurostream that can, in just a few minutes, in a fully automated way, exactly identify and quantify the volume of the lateral ventricles, which we've found is a really good marker for overall brain atrophy. And we're hoping that we can use that to track individual progress or response to therapies, for example. Another one that we've worked pretty hard on is a tool called Deep Gray, which is a tool to measure thalamic volume. And I mention this because it's, I think it's kind of impossible to do science these days without hearing something about deep learning and artificial intelligence. And it has its upsides and its downsides, but it really is a game changer in a lot of ways. And so we developed an AI-based tool to quantify and detect the thalamus, which is a central brain structure that acts kind of like a major relay connecting lots of different brain regions and different inputs together. And it's often kind of like a canary in a coal mine that can go first in disease and can be a good marker for seeing for diagnostic and prognostic purposes. And so we use some deep learning, and I think it's kind of fun to watch it train. The neat thing about deep learning is that it's a little bit different than traditional computer vision and other machine learning techniques, in that those techniques, we tell the system what we want it to learn, we tell it what to do, we tell it how to figure things out, which is nice because we know what it's doing. It's not a black box. On the other hand, deep learning tools, we don't tell them how to figure things out. We just tell them what we want them to do and let them figure it out from the data. And that has some downsides. It can be a black box, but the upside is that it can also be very unbiased. So it figures out its own inputs from the images, and it's not restricted to what we think might be important in classifying a thalamus. And so you can see kind of this is a baby AI here that's just starting to learn. And we keep training it and teaching it on other images to locate this structure here. Again, this is called the thalamus. Excuse me. And we train it some more. It's kind of adolescent now. And it's pretty neat. You can see it start to understand a little bit what a thalamus is and start to really lock on and learn the thalamus. And it even does a good job of respecting this structure that's called the internal capsule in the brain. That's actually a separate tract of white matter. So I think deep learning is a pretty powerful tool. It also runs in half a second, quarter of a second actually. So it's the same technology that's allowing things like Snapchat filters and Facebook to detect faces and track things is also allowing us to do similar things on medical imaging. So that's kind of one broad area. Another broad area is tissue measures of disease. So we talked about, for example, those lesions, and again, I want to emphasize, don't underestimate the power of quantifying things. For learning and for research. And so one good tool is to use multimodal imaging to fuse those together and to delineate the exact edges of pathology, so that instead of just having a radiologist look at that pathology, we can align it together over time, quantify it, and say that's 2 or 3%. And that kind of approach can be really important for a couple different reasons. It can be important for clinical trials where we want to understand the impact of a therapy. In a lot of these diseases, we still don't have perfect cures yet. And we're in a situation where we want to compare different potential drugs and understand whether one's working better than another. And it may be a case of 2 or 3% difference, but we want to see that. That 2 or 3% is incredibly meaningful if it's you and your brain, and you want to know which drug is preventing or limiting the speed of expansion of these areas of pathology. Another area where we can look at tissue pathology to understand disease, another MRI technique that my colleague Ferdinand Schweser here is really an expert in, and that we've done a lot of work together with, is iron quantification. So because MRI works fundamentally on magnetic principles, there are some neat ideas that have been developed to image the magnetic field of iron and identify areas of concentrated iron, and we can see iron deposition in vivo and understand where iron is going. And that's interesting, I think, for a couple of reasons. One, because we see iron dysregulation in a lot of pathology. We have a number of really good people at UB researching iron. And another reason I bring it up as a point is because it's a little bit more of an exact thing that we know this is iron. As I said before, we work a lot with things like T1 and T2, And it's nice to have some methods that tie more directly to our macroscopic world. Another technique that we've developed over time and worked with a lot is called voxel-wise MTR. And this is a method for imaging demyelination and remyelination in vivo. And there's a lot of neurological diseases that can involve demyelination. And again, one of the major clinical things we want is to understand whether drugs can prevent demyelination or promote remyelination. And we want to also understand how those processes are occurring in the disease from a basic science perspective. Do they occur? What's the timeline? Do they occur in the same regions as ongoing damage? And so this is, I think, an interesting example, because I'll show you 3 different people, and these yellow outlines here these are people with MS, and these yellow outlines are lesions. And what's very interesting to me is all 3 of these people have the same overall lesion load. So they have the same volume of lesion in the brain, very similar disease duration as well. But this person has a lot of this green over time, which is remyelination. This person did very well clinically. And this person had a more middle of the ground sort of trajectory. And you can see that they have kind of mixed a lot of remyelination outside the lesions and still some ongoing demyelination in the lesions. And then we have this person here who has substantial demyelination throughout the lesions and some outside as well. And so the point is, classical MRI methods, they would just see that volume of lesions and say these 3 people look the same. And when we start to use these more sophisticated tools to look a little bit deeper, we see that there could be substantial differences. So I think this is interesting and important from an imaging perspective, and also for everybody who's investigating these kind of things to start to answer these questions of why, what's different about these 3 people. So the imaging can let us also identify groups that we can then potentially hand off and say, what's different about these people? Maybe the geneticists can take a look and understand if there's a genetic predisposition for something. So those are that's a brief overview of some different ways we look at different tissue measures of disease. One of the things that we've been really interested in from a how the brain works kind of perspective is structural and functional connectivity. And this is pretty neat stuff. I think the brain is broadly made of a couple of different parts. For those of you who don't have a neuro background, the key organization of the brain really is that the gray matter here, around the outside, this cortex and the deep gray matter here, really contains most of the cell bodies of the neurons. And the thing that I have to say is always surprising to people who aren't in neuro is you always learn when you're in grade school that cells are very tiny things. And for the most part, that's true. But neurons, actually, they're very tiny in 2 dimensions, but not in the 3rd. Neurons can actually be up to 10 centimeters long in some places, a single cell. They're so they have the cell body, that's the head, and then what's called the axon that acts almost like a wire connecting it to other areas of the brain. And those are macroscopically long, very long. And so what we see when we look at a brain, when we cut into it or do conventional imaging, you see the gray matter on the outside, and you see this kind of nondescript white matter. This white matter is all those axons, but we can't really unlock it. We don't really see what's the organization of it. We don't know whether this is connecting to here or connecting to somewhere else, or going to here. We can use a relatively newer tool called diffusion imaging that lets us actually take each little piece of white matter and identify how the water in it diffuses to get the orientation of white matter, of the tissue there, to get the primary orientation of those wires, those axons. And so we can take this, what looks like homogeneous mush here, and we do that imaging, And we see that there's really this incredibly beautiful structure of the brain that's revealed, actually even better with imaging than it is with histopathology. Because even when you cut into a brain, you still see just that massive white matter. When we use these diffusion techniques, we can start to really tease out all these little fibers and see that it's not random. It's like a beautiful wiring closet that connects everything in very specific ways. And we can start to identify all those tracks and quantify them. So, you can see here, for example, this is called the corpus callosum, the major area that connects the two different hemispheres of the brain. These are the cortical spinal tracts traveling that bring sensory motor signals to and from the sensory motor cortex down to the spinal cord. You have some visual tracks going in and out of the screen here. And we can start to really see those in vivo, in a real person. Any of you could go sit in a scanner for 10, 15 minutes, and we'd be able to see that in your brain. And so what we can do with that information, lots of things. But one thing we do a lot of is parcellate the brain out like we looked at before. But then instead of trying to get the volume of all these different brain areas, we can use that tractography. This is a busy slide, don't worry about the exact details, but essentially what we can do is quantify exactly how connected, how many fibers connect any two different areas of the brain. And so we can build up what looks like alien hieroglyphics here, but really this is what's called the connectivity matrix, where we have all, let's say 86 brain regions here, and by the same 86 brain regions here. And what we get is this kind of heat map of which areas are connected. So the brightness of the pixel tells us that this region up here is strongly connected to this region here, whatever region we're looking at. And what's really powerful is that this can be an individual kind of fingerprint of brain structure. I know Ben was talking about schizophrenia a little bit before. This has been used a lot to understand whether there are potential wiring issues. We use it in MS a lot to understand, for example, when we have a lesion in a different area in the white matter, it can have very different impact depending on where it is, because it impacts and intersects different areas, different cables or different fiber tracks that are going to different places. And now we have tools using this diffusion tensor imaging to actually quantify that. And we can build up maps of exactly on an individual basis what regions are disconnected by disease. And that helps us monitor individual patients. It also helps us ask basic science questions about what happens, what regions are important for what sorts of outcomes. So we did one interesting study with my at the time PhD student Tom Fuchs, who did a lot of incredible work, and he looked at personality traits, conscientiousness, and how they were affected by lesions in different areas. And he was able to actually map out the specific sets of regions and their connections that were able to predict that. So I think that's interesting from a basic science perspective, because it tells us a little bit about how the brain is organized. And it also lets us understand, prognostically for individual patients, what lesions in different areas potentially mean. The last major thing I want to mention is functional MRI. So pretty much everything we've talked about so far is what we'd call structural. It has to do with the physiology, what you would see if you were to cut a brain open. But of course, the really interesting thing about the brain is not just what it is, but what it does. And fMRI is just an incredibly cool technology that lets us see the brain in action. And it shares a lot in common with techniques like EEG, for example. It's a bit slower than EEG, but it has a lot better spatial resolution. So we can see things. We can actually put you in a scanner, let you sit there for some time and ask you to do things, or, for example, play music or sounds and then not sounds, and see which areas of your brain light up differently. So this is an example of what happens if you put someone in a scanner and give them auditory stimulation. And you can see the temporal auditory cortex here just start to light up. So this is a really powerful tool for mapping brain regions, and lots of investigators have gotten a lot of value from it. I think one of the neatest things is resting state fMRI, is that even when you put someone in a scanner and don't have them do anything, we find that the brain just naturally kind of segregates itself into separate networks that tend to work together. So the brain is really built of kind of special teams that work separately, and they correspond to some really well-known networks. So you have these key visual areas. These three networks are very visual. This is a very interesting one that is still an area of active research called the default mode network that tends to light up when we're not doing other tasks or when we're having introspection, thinking about ourselves or more abstract things. Cerebellar networks, this is sensory motor. This is that auditory one we looked at. And these, what's really interesting is these come out naturally. So that tells us something about the brain. And we've used that kind of intrinsic connectivity of the brain to look in disease as well. So we have looked at resting state connectivity of the thalamus, again, those thalamic regions, and tried to understand how it can tell us about the kind of dysfunctions that can lead to things like cognitive dysfunction or fatigue in neurological disease. This was a study we did in multiple sclerosis where we compared MS patients and healthy controls in yellow, MS in yellow and healthy controls in blue, and looked at the overlap in red, and we can see areas between these different thalamic nuclei that are correlated with different outcomes. Another thing we were able to look at, and that I think will be very interesting in the future, is understanding, does this functional fingerprint of an individual, how their brain is wired up and how it functionally connects, is it predictive of response to therapies? And so we ran a study on cognitive rehabilitation in MS, and we found that default mode network, the connectivity of that at the beginning, at the baseline, was a strong predictor of who was going to respond well to that. So it can potentially help us triage people into different types of therapy. So I think those are kind of the main things. I think some of there's just an incredible arsenal of tools with MRI. We've got thousands upon thousands of data points now, and we need more people like yourselves to come and get involved in the field, start analyzing these things, start making connections. I want to end on one cautionary slide because we're getting in a period of big data, and I think the big data is incredible. It's letting us do amazing things in terms of prediction, in terms of putting things together. But we have to be very careful to use our statistical common sense and to be careful not to see patterns where they don't exist. And that's one of the places that the scientific method has been very important, that we need to make sure we still have falsifiable hypotheses and test them. And this is just a kind of fun example. I like to show that these people, these things are just objects, but I'm sure most of you see faces when there are no faces. And when you have 1,000 data points, you start to see connections, but some of them are real. And so the challenge is to get enough people working together to tease out those real patterns. So I think that's it for me for now. I'd be happy to discuss anything anybody wants to talk about or answer questions. Just stop sharing.
Be your one lone auditory clap.
Thank you, Ben.
I just want to throw in really quick while I have the chance. I think it's so cool that I think the coolest part about your research is that you're taking this system of brain imaging that's already ubiquitously implemented in medicine, and you're finding ways to make the information even more valuable, which is, I think that's just so cool. Like that's the ultimate translational.
It's very cool stuff, and I think very, very applicable. I mean, I don't want to say anything. There's a place for everything, and that's, I think, one of the coolest things about science for everybody here is that it's really big. There's whatever you're interested in, you can make a difference and do something. So basic science is still vital. It's what makes the major leaps. But translational science is also vital, being able to take these things and move them. Let me just see here. I see some Yeah, the technique for prosthetic limbs, that's there are definitely people doing that, that kind of work, with fMRI. But the problem with fMRI is you can't walk around with a with an MRI machine. Um, so one of the things that, it's not my area, so I'm not an expert in it. Take what I say with a grain of salt. But I know that there are, for example, challenges where people build machine learning tools where there are data sets where they've shown people different objects. They don't tell the machine learning people what the objects are. They just show them the brain activities, and you have to be able to guess from the brain activity what that person was looking at, for example. So I think that the prosthetic stuff will probably get the most practical thing will be EEG. But the problem is that EEG has terrible spatial localization. So, what I would envision is probably that we'll see EEG that's informed by what we learned from fMRI to kind of constrain solutions. One of the big challenges in EEG is what's called the inverse problem, where you try to figure out from the signals you get on a scalp where that electrical pulse was coming from in the brain. And it's not a 100% solvable problem, but you can get a solution that's probably good enough to help a lot of people. Let me see. What was the transition like from your computer-based background to being able to integrate it with neuroscience? So, like I said, I had a pretty interesting path. The way I got into all this was almost by accident. When I was in undergrad, I had a friend who was in med school at the time, or pre-med, and he started working in a neurology department. And they were trying to do things with computers, and I was doing my computer degree. So I came and just started helping them with basic IT stuff. And then kind of really started to like it, to the extent that that's what I, started doing my master's and my PhD work, especially my PhD in. So it was almost a backwards thing for me, that I kind of, I was always interested in CS. But I think computer science is very integratable. So if that's what you're interested in, I guess my advice would be have applications. For me, there's theoretical computer science where you really focus on algorithms, which, like basic science, is very important too. But for me, what I really liked was having the application and being able to see the computer science techniques, the computer vision, actually be able to be used to make these real kind of draw these real conclusions from medical imaging data.
So one question for the actual image generation.
Sure.
Are there multiple NMR techniques being used at the same time for the data collection, like HNMR and CNMR?
So yeah, so basically, it's in a clinical MRI machine, it's all hydrogen. Like everything you're seeing in those images, 99% of the time, is coming from hydrogen. There are some interesting techniques. There are, in the research arena, things that look at sodium imaging, fluorine imaging, different things, but all the clinical work, everything is done on hydrogen. It just happens to be that you can learn an awful lot from hydrogen, since it makes up 67% of the body. There's water everywhere, basically, and as long as you can ask water questions about its neighborhood, you can form a pretty reasonable picture of things. That being said, it's not always water. So we do techniques like magnetization transfer, for example, that basically looks at the difference between the way macromolecules will have a very different absorption range than water. Water has like a very wide absorption range, and macromolecules have a pretty thin one. So we do exploit those kind of things to make some unique imaging types. But yeah, almost all hydrogen. Yeah, good question.
Thank you.
Most excited about for the future of the field? Yeah, I mean, I think So in the short term, I think the thing I'm most excited about is seeing these tools start to be translated to be helpful for real clinical applications. You know, to see people start to use brain atrophy tools and quantitative methods to be able to make decisions. The imaging field has been kind of simultaneously very quantitative and accurate, but also on the clinical side has been very qualitative. You know, when we send people for a blood test, you get these really nice quantitative things back that tell you exactly what your levels are for all the different, you know, things that are there in your blood. When you send somebody for an MRI, you tend to get back a qualitative review from a radiologist that just says, looks like there's maybe some new pathology and some growth. So I think, you know, and that's, you know, to a large degree because computer vision is really hard. There's some hilarious quotes that I've read in the computer vision literature that, you know, a lot of the original founders, you know, the earlier people working on it in the 60s thought it would be solved, you know, as a grad school project in a summer, that computers would be able to figure out vision. And here we are 60 years later, and it's still hard. But deep learning has made a big difference, actually. So it's getting better. Long term, what I'm excited about is probably more the stuff you guys have been asking about. You know, I see some questions about human brain interfaces. You know, obviously I'm a huge sci-fi fan, so you know, long term, I think the potential to directly integrate computers somehow with the brain would be vital. I don't know if that'll be with MRI, because MRI is a giant bulky thing, but I think we make a lot of strides, and hopefully it'll be smaller. You know, I think that, I don't know if I'm probably too old, but you know, Star Trek, if any of you watch Star Trek, they always had the tricorder that they just do the scan on quick, and we have that now. Like, we have a tricorder from Star Trek. It's just a lot bigger right now. But if you looked at computers 30, 40 years ago, they were a lot bigger too. So, you know, hopefully if we make it small enough that it can be used, long term, that's what I'm excited about. Short term, excited about direct clinical applications though. Deep learning, book recommendations. Ooh. I'll be honest with you, the problem is deep learning is evolving so rapidly that by the time a book is published, it's out of date. You know, that's the problem with a lot of the medical field too right now, is, you know, you buy a textbook and we already figured out it's wrong. So I wouldn't recommend a book. If you're interested in deep learning, there's a great course called Fast AI by Jeremy Howard that's really practical and very accessible, and it gets your feet wet really quick. You know, I think you do need to eventually spend some time with the math and get the fundamentals, but if you want to just learn a little bit, Fast AI is 100% the way to go. Yep, that's the one. It's and it uses PyTorch, which is way more accessible than TensorFlow for people who are into that kind of thing.
I just figured I'd type that out for anyone interested. While we have a break here, I'm just going to take this opportunity to show everyone something cool. This right here is I don't know if you remember this. This is a image of my brain that was taken as a part of the class by Dr. Dwyer. And you know,
yeah.
what more relevant? I'll never have an opportunity where this is more relevant to show someone.
Yeah,
so.
show it. I still look at mine too. It's really, it is an amazing thing that we didn't have to cut your head open to make those images, right?
Right.
Yeah,
my brain's there and it's still here, which is awesome.
Yeah, it's really incredible technology, and it's getting better every day. You know, that's what I was trying to get at the beginning. I'm very excited also about MRI. I guess the third thing I would say I'm excited about is just the new contrasts that'll come from MRI. It's really versatile because, you know, like I said, you're not it's not a one-trick pony like X-ray or CT is, you know, like not to denigrate it, but CT tells you one thing. It tells you how much does X-ray pass through this tissue, and you can get a lot of mileage from that if you're clever and creative. But MRI is really kind of starting a dialogue with the protons in the brain, and you can ask all those different questions and learn all kinds of different things. So it's a lot, I think, more versatile. It's also, unfortunately, a lot more expensive. that's the downside. But yeah, it's a pretty neat thing. Oh, what's your favorite computer language? That's a good question. Python is the lingua franca of science at this point. I mean, if you want to learn something, learn Python. I mean, I've worked in a bunch of different languages. I think it's really valuable to know different languages because the most important general advice I would have for people is probably to try to learn different tools. You know, because I've seen it happen to myself and others too. You know, you get really good at a tool and the tool starts to constrain the way you think. So, you know, try to get different tools in your tool belt so that the science can guide how you're thinking and not the tools you have. You know, it's you have a hammer, everything starts to look like a nail kind of thing. So, you know, learn multiple languages. I would certainly recommend learning a functional language because it just bends your brain in weird ways. But then Python is what you're going to get 90% out of. And you know, if you're interested in, you know, if you're, I mean, everybody's here because they're into science. So I would say R actually it's a little bit of a weird answer because it's not a general purpose programming language. It's a statistical programming language, but I really think that's honestly statistical literacy is one of the most important things you can do to be a good scientist. You know, we all have, it's like that picture I was showing at the end. We all have incredible pattern recognition systems in our brains, but they evolved such that we find patterns more than we don't. You know, it's always a classic thing. It's better to think there's a tiger and run away and live another day. So we're kind of biased to find patterns, and stats is the best way to prevent yourself from making wrong conclusions. So I would say Python and R. Oh, I don't know the answer to Andrew's question. That's a complicated one. Parkinson's is not my main area of specialty. You know, we've certainly, I can tell you from an imaging perspective, one of my colleagues has been working a lot with a method called free water in the substantia nigra that's been pretty interesting as a marker for those areas, and to understand how the cellular structure is degrading over time, and it's been a pretty good technique, but I couldn't comment on the specific study. In which aspects does the research with MRI have to improve to translate that to medical care? You know, I think one of the challenges, we do a lot of our scientific and research work, and I mean we as a you know, the whole scientific community, on big groups of people, you know, and then we do our t-tests. And what's more important from a clinical perspective is not whether you see group differences, but what it means for an individual person. So one of the aspects that we see problems, for example, is, you know, let me put it in context for you. So this brain atrophy stuff, you know, a person who has MS can potentially have a brain atrophy rate of up to 1% per year. The normal rate is closer to 2 to 3%, 0.2 to 0.3% per year. So that's 3 or 4 times the normal rate. And on a group level, it's incredibly important and it's a lot of tissue. But when you have measurement error of even, let's say, 1%, you know, and you're trying to track something that's changing by 1%, it starts to become very meaningful on an individual basis. So I think having those kind of normative ranges is pretty important. Having what's called like confidence intervals, but meaningful change values, things like that, are very important for translation. When you do registration between brains, you typically apply an Fm registration. Do you find that there are anatomical artifacts in the structures when you do this? From Steve. Yeah, that's a good question too. It's a complicated answer. It depends on what you mean by between brains. If you mean between individuals, then I would probably do warping with something like ants. If you mean between brains of the same person at different times, I would do like a rigid 6-parameter registration.
I mean between different patients. So,
Yeah.
so whenever you're warping things, ultimately my question is, because it's important for statistical shape modeling, and we had talked about this before, trying to understand the differences between structures. Whenever you do warping, isn't that changing the anatomical structure itself?
Oh, I see what you're talking about. Yeah, that's a great question. So cleverer minds than me have also had that question, have answers to that question. And there's so one of the major ways to deal with that is something called Jacobian modulation. So without getting too mumbo jumbo-y, basically when you do that warping, you can actually also track how much you needed to, because so like you said, when you do a normal registration, you're just moving things around, you're aligning two brains. When you warp things, you're stretching and smushing it like silly putty. You can mathematically keep track of how much you're stretching and smushing it, and encode that in something called a Jacobian determinant, which is basically saying at each little pixel, at each region, how much did I have to stretch or expand to do this? And then it depends on what you're trying to do, but you can modulate your statistical test by that, to correct for it. So you can correct. I mean, there's no free lunch though. You have to decide whether you want the anatomy to match spatially or whether you want to, be sensitive to those kind of things. So it changes whether you're talking about concentration or absolute volume if you do that. But you can certainly control for it. And it's a pretty. I could send you some literature on it. It's a good method for handling what you're talking about.
Okay.
What sounds like a dance move? Jacobian modulation. Yeah. It does. Any other questions? People, ask questions. Never feel stupid asking a question. That's the number one thing. The other, I've always found in science, it's not what you know, it's what you know that just ain't so that always gets us in trouble. So it's always better to ask questions and even about things you think maybe you understand, ask the questions and find out, and don't be embarrassed. Nobody knows everything. That's the beauty of science.
Actually, I have one more question. So whenever you do your analysis, do you just get do you get the raw data from like the sinograms that are generated, or do you get? I'm assuming that they're still sinograms because it's a tomography, but
yeah, it's not quite a sinogram. It's a little different the way MRI works. I mean, you're on the right track though. We can use raw data. For us, it's actually the way MRI works. I mean, I lied about some stuff earlier a little bit to make it simple, but it's it gets encoded in what's called a frequency space, K-space actually, which is like a Fourier encoded image.
okay, so like a Fourier transform.
Yeah, we do an inverse Fourier transform basically to get the image. So when you actually acquire it, you're actually acquiring things at different frequencies by modulating the frequency a little bit. And so you get this thing that's basically the Fourier transform of what a person's brain looks like. We get that back out. Sometimes it depends on what we're doing. For like the iron imaging stuff, we do have to use the raw data, and we use it quite frequently. A lot of the times the manufacturers themselves though have put a lot of work and validation into the reconstruction methods, and we don't need to use the raw data for a lot of these things. We use the more standard DICOM types and work at a higher level. But sometimes you need the raw data. Yeah, I would definitely recommend getting it.
And I'm assuming that you guys still use like a medical physicist to evaluate all of the artifacts and stuff like that that are present within your equipment.
Oh yeah. And I mean, we use the clinical equipment, so it's always, it's always ACR accredited and stuff too, and they have to go through all the different tests, and it's that's the interesting thing about doing clinical research is, you're also subject to a lot of regulations that can be both very good and also make things difficult. but it's there for a good reason.
Can I ask a quick question? So after hearing that exchange between you and Steve, so Steve is doing research that's quite similar to yours, or at least somewhat related, where, just hearing you guys talk, I'm sure a lot of people here are wide listening and thinking, I have no idea what they're talking about. I think when you look at, something like all the knowledge you have about brain imaging and all these different, technical approaches, I feel that students just sort of beginning their journey may look at that, or whatever their field is, requires them to learn and say, how am I ever going to understand all this? Do you have any sort of advice or anything, any comments on just like how that process goes, and maybe any encouragement for students concerned about that?
Yeah, no, that's a really great point. And I think, it's hard not to use jargon sometimes, because it saves time, once you know stuff. But don't be afraid. It comes iteratively. Learning is a circle. You learn a little bit, then you go back and revisit, and you fill in the gaps, and then you go back and fill in the gaps more. And I mean, I had no idea about this stuff when I started either. Nobody does. Nobody's born being a great scientist or knowing these things, or understanding everything. And I mean, I still read papers and don't have to go look things up. I get stuck in a Wikipedia hole all the time. I mean, it's the nature of the beast. You never know everything, and that's the joy of science. But it's also, a little bit the curse that however deep you go, there's deeper. so yeah, don't be afraid of that. Never be afraid of not understanding stuff. I think the most important thing I could say is to go slow and take your time. Because of exactly what Ben's saying, you can get worried and try to catch up and go fast, but that's the exact wrong thing to do. It's more important to take things slowly, make sure you really understand things, build a slow foundation, and build on top of that, because otherwise it all comes crumbling down. Learn the basics, learn the next pieces. Somebody was saying, I wonder the same thing. Somebody coming from a psychology background is just getting into science. And yeah. Yeah, I think it's general to science. I think it's, but I mean, I don't even think it's specific to science. I mean, my wife got into knitting lately, and I'm like, what? How are you even doing that with your hands? Like, I mean, she's creating these complex patterns, and it's just, we're very adaptable creatures. You learn over time, and you build it up one bit at a time. It doesn't come as one big piece. So that's the thing. Don't be afraid. Have some faith in yourself, and the most important way to learn is to not be afraid to look stupid. That's the most important thing. Don't be afraid to look stupid, or you will never get anywhere. And then somebody's asking, is the human brain connectome? Yeah, the human brain connectome project, HCP project is amazing. They're doing great stuff. They have some specialized scanners. They've made their data available. We're using their data a lot, actually, for some of those tractography techniques. It's a great thing. It's multi center, people putting together data. They're studying all kinds of stuff, and building, the genetics, the aging, all the different components, and the functional and structural imaging. It's really great. I would love to see more initiatives like that, with really team science. Are there any techniques emerging that may render MRI obsolete in the next 20 to 30? I mean, Bill Gates said that nobody would need more than 256 kilobytes of RAM. So it's impossible to predict the future of technology, especially now. We don't even know. How do I know that there's not going to be some neutrino technique that we don't even know about now? I would love it. I mean, I think it would be great if we made it obsolete, because right now it's it's amazing technology. It works, but it's still very expensive. You need this giant room-sized superconducting magnet. So if there could be a tool that would let us do similar things, cheaper or smaller or better resolution or any of those things, it would be great. I mean, I don't see anything on the immediate horizon that I'm aware of that is very similar to MRI in terms of what it's capable of. I've seen some work on miniaturizing MRI. They've gotten to the point where they can do some decent things with like hair, the old salon style hair dryer size MRIs. But I don't know. It's a great question, but I would not want to wager on one way or another.
In your research, have you ever done cross-modality inference, like between, for example, PET and MRI?
So we haven't done PET MRI. We've done a little bit with EEG and MRI, and we've done pathology and MRI quite a bit a while ago. It's funny you asked that though, because we're actually just started a big PET study that we haven't actually got the data for yet, but we're going to be doing that exact kind of stuff. We're working with a new tracer that some other people developed to look at microglia in the brain. So, those are the kind of a glial cell that kind of is part of the immune system in the brain and can activate and can be potentially part of some of these diseases, particularly MS. So there's some new tracers for something called TSPO, and we're using we just got the tracer working. It's hard to work with all the, I'm used to this nice non-invasive MRI, and working with PET tracers where you have all this harmful ionizing radiation is a lot more difficult. But I think it's a good question. I think there'll be, multimodal imaging is definitely where things are at. The thing I like about MRI is that it's multimodal in itself. We get 10 different types of images with different resolutions and totally different things. Anything else? Other questions?
Got one more.
Great questions, great audience.
Somebody asked a question on the chat real quick.
What was it? Did I miss it?
It's the bottom one by Brooke Montgomery.
Oh, sorry. Is there anything in these CS and brain imaging ideas that could be related to studying cognition? Oh, that's a great question too. Actually, that's been a really specific area. I didn't really have time to talk a lot about it, but we work really closely with a neuropsychologist, Ralph Benedict at UB, and that's actually what Tom Fuchs, the PhD student I mentioned, did a lot of work with cognition. So absolutely, especially that connectivity stuff, the functional and structural connectivity. I think one of the most interesting questions that we still don't fully understand right now is we see, there's this concept of cognitive reserve where we see that some people who have very similar degrees of pathology can have very different outcomes. And I would hazard a guess that some of that is because we aren't distinguishing the pathology well enough, but there's a lot of evidence that a good part of it is also due to the fact that some people's brains are somehow able to work around that pathology more flexibly or in some more efficient way. And we've seen some evidence of that with this, functional connectivity. People who have the same structural connectivity deficits, but are somehow able to maintain similar functional connectivity. So, we don't understand exactly what the mechanism of that is, whether it's some kind of, alternate routing or whether the same systems are somehow working harder or better or something. And so I think that kind of unlocking what that cognitive reserve, we know things that contribute to cognitive reserve are things like, enriched environments, exercise, reading from an early age, all that kind of stuff. But we don't really fully understand what the physical substrate, what's the actual mechanism going on in the brain that's allowing that to happen. We don't understand whether it's something about the wiring or something about the functional at a higher emergent scale. Like we don't even know what scale it's it's at, whether it's at a closer to the hardware or closer to the software. So I think, yeah, a lot of questions in that regard. I don't know a lot about electron imaging personally. I wouldn't know enough to guess. Sorry, it's a good question, but I don't know enough to comment on it in any meaningful way. Okay. Well, I actually have to go make another call. But it's been really great talking to you guys. You know, and I think Ben and Steve have my email. If anybody has, you know, if anybody has questions, I'm more than happy to discuss or answer them. You know, it's really great to see you guys doing this kind of thing. Ben for organizing, Stephen for organizing. You know, it's a big field. So never feel intimidated. Never feel like you can't do it. It takes time, and you get better with time, like everything else. And I think from my perspective, it's a fun journey. You know, have fun, learn while you're doing it, and it's extremely rewarding because you get to learn things about the world, and they tend to be very meaningful. So I wish you all the best of luck in your future endeavors, and never give up. Okay. Bye, guys.
Thanks, Dr. Dwyer.
Thank you.
Thank you so much, Dr. Dwyer. Lots of great advice and great information.